Welcome. My name is Marty Leon, and we're sitting in the TCTMD studio, and we're going to have a fascinating discussion about the DurAVR THV system. It really is going to be a discussion about some of the things that are happening in innovation in valve leaflet technology that really can affect our clinical outcomes. I am surrounded by some pretty awesome people, and I'm going to introduce them first before I give you a quick introduction. To my immediate right is Chris Maduri from Karolinska in Stockholm. Next to him is Becky Hahn, who's an interventional echocardiographer who works with us at Columbia and has made a career of studying valve leaflets, at least as they're being applied with new technologies. Next to Becky is Thomas Modine from Bordeaux. He is a cardiac surgeon who is acutely aware and interested in valve leaflet technology and has worked with us in transcatheter projects previously. And finally, Mike Reardon, who probably has as much exposure to more different transcatheter heart valves as anyone in the world at this point, a prestigious cardiac surgeon from Houston, Texas. So with that group, I'm going to give you just a short introduction. I would say over the last 20 years, we have focused on being able to develop what is a consistent, predictable, simplified transcatheter heart valve technology that allows us to treat most of our patients. We've been very effective in focusing on the index procedure and talking about how we can improve the procedural steps, on how we can make the procedure simpler, on how we can reduce clinical outcomes, adverse outcomes. So certainly that's been highly successful. We've spent perhaps less time thinking about what happens before we do the index procedure, which we call the upstream effects, and then downstream after the index procedure. First, upstream, we're now beginning to explore patients that really aren't so symptomatic, where we're looking at the potential of preemptive AVR to be able to change the natural history and improve clinical outcomes before patients develop symptoms. And then downstream, we're talking about what is the lifelong journey of these patients who now are introduced to an implant. Now, an implant is really an iatrogenic stimulus for potential negative outcomes. So we have to be thinking about hemodynamics, we have to be thinking about durability, we have to think about the possibility that we could have one valve that would last for the lifetime of a patient. So these are some of the considerations that we're going to be discussing, and the upstream and downstream effects are intimately linked. As we do more preemptive treatment of younger patients that are lower risk, these valves do need to be better functioning, better hemodynamics, treating all patients, and more durable. So this is really the focus that we want to get into and really point to what we think are some of the critical features of valve leaflet technology that does impart specific characteristics that can modify the natural history of valves and potentially improve clinical outcomes. So we're going to have a series of discussions. After each discussion, we'll have an interactive a conversation about some of the observations, and hopefully we'll, as a group, come to a consensus as to where we're going, particularly as it relates to some of the new technologies. And we're going to begin with Chris Maduri, who's going to spend some time discussing some of the interesting aspects of the DurAVR THV system and really speak a little bit about the importance of technology on valve leaflet design.
Thank you, Marty. So I think what you alluded to, Marty, is it's right where we are as a field right now. We are incredibly effective at providing an absolutely wonderful index procedures we've seen across the low-risk data even now to date, and we continue to see really great outcomes. But that's been our primary focus, and now as we're starting to move into expanding risk populations of lower and lower, younger and younger patients, earlier in the disease process, we have to be thinking about not just the index, but the longer term implications, I think, of the valve that we implant. And I think right now, we're kind of struggling in the aspect that we are often thinking of compromises when we think about optimal lifetime platforms. Now, I don't think that things really live in the buckets of just balloon expandable and self-expandable, but that's kind of the framework that we often are thinking of we think about the trade-offs in decision making. And often when we look at balloon expandable, we think about things such as a short frame height, which allows ease of coronary access and potentially facilitates future valve and valve interventions being easier. And additionally, it's an incredibly easy and consistent procedure. It's built off of the backbone of doing balloon expansion to get a stent somewhere, which is the foundations of interventional cardiology. And so it's relatively easy to translate around a mass of users. But on the other hand, we have something in the self-expanding category that's incredibly important, likely, for optimizing lifetime, and that is optimal hemodynamic performance. And we know more and more, and Becky's done incredible work showing us this, that there are real implications to patients that don't have immediate optimal hemodynamic performance. And additionally, the field is becoming more and more excited about this idea of commissural alignment, which I think, at least at this time, is more effectively done in self-expanding platforms. And so I do believe that this is an area that DurAVR, as you'll see from unique leaflet technology design, is potentially specifically advantaged to provide a taking away that compromise in an optimal potential strategy. And that's because it provides this shorter frame height with an ease of use of the balloon expandable platform on a cobalt chromium stent, but also can offer, I believe, optimal hemodynamics in commissural alignment. And through those things, I really believe this can offer the best of both worlds. Additionally, there are things that we'll talk about today, I think, that are really pretty interesting with potential future implications we don't even really talk about much. What is the implication of having almost normal-like flow dynamics? What happens when the leaflets are much more like the native human valve? And additionally, there are implications on the tissue itself, which is an advanced acellular tissue that we think will lower the immunogenic response. And so, for context, what DurAVR is, it is unique in that it is not 3 pieces of tissue. It is a single piece of bovine pericardial tissue that has been shaped to mimic the native aortic valve, as you can see in this image here. And this has many, many implications, but I think the first and foremost to remember is this is the way the native aortic valve truly looks like compared to traditional 3-piece valves. So some of the potential implications are around just the functionality itself. We can see in this image here that we looked at traditional 3-piece valves, you can see that there's a significantly longer coaptation length. And additionally, because of the lack of 3 pieces being sutured, it actually allows for a significantly larger effective orifice area, all of which have long-term implications. And so, in summary, I do believe this is a valve that has been optimally designed for lifetime management. It provides with the backbone of a very effectively proven tissue that has been used in over 20,000 patients now to date. It has a unique single-piece tissue design that I think provides optimal hemodynamics and more native performance, large open cells for optimized coronary access, a delivery system that allows easy rotation on a balloon for optimal coronary alignment, that we really are looking at a technology that potentially can help modify the trajectory of some of these lower risk patients as well as the current patients we treat.
Thank you, Chris. That was really a beautiful description of DurAVR and some of the differences. So if I were to break it down, clearly the tissue is different. It's been treated in such a way where the assumption is there's going to be less from the standpoint of structural valve deterioration and calcification, that it has the potential to last longer.
Yes.
So how do we demonstrate that in vivo?
Those are great questions, Marty. I think one area obviously is that there's a lot of stuff that has to be done outside the body now because we don't have the time horizons. I think the idea that just putting something on a pulse duplicator and running it for 1 billion cycles is probably not the effective strategy because there's so many other implications. Obviously, we want to look at actually the calcification process and head-to-head testing just across the tissue itself. But there's people like in Vancouver, Stephanie Sellers, who are doing remarkable work trying to get early insights to help guide us early on, of which, talking to Stephanie, some of those signals of early things that need to be vetted out more effectively, we start to see here. One area I think that we need to start thinking about as a field is after several years of these valves being implanted, doing early PET tests to try to identify, are there early signals of uptake of inflammation, durability issues, to try to translate that clinically so that we can identify potential early issues earlier. I think fortunately here, the foundation is in acute hemodynamics, and it has as much as anybody else almost today with the longer term, but we need to really start working as a field to identify these things more effectively.
Yeah, I think that's going to be very important, the innovation in cardiac imaging. And some of our UK colleagues have been working with sodium fluoride PET to be able to look at microcalcification. And those patterns are fairly interesting and probably will predict earlier valve deterioration. So hopefully there'll be ways to validate that. I'm very intrigued by this 3D valve tissue concept. We have toyed with this for a while ago, and maybe I'll ask Thomas, why hasn't that been incorporated in surgical valves?
They didn't have the idea probably at the right moment. I mean, most importantly, I think one very important subject for us as surgeons is to be sure that we are delivering the best and the optimal therapy for the patients. And that's why we have developed surgical techniques which were very difficult. The Ross procedure, enlargement of the annulus. So we have been always paying great attention, I would say, to optimizing the flows and the hemodynamics. And I mean, I think there is always a trend in research. People try to follow each other, and you need sometimes someone that come and say, we have to look differently and do it differently. And this is why DurAVR is a new concept, which I am sure that it will have major implications in the future of this therapy.
These surgical valves were designed many, many years ago, and we've had very little competition other than other surgical valves.
Yeah.
TAVR valves has now fundamentally changing the way surgeons approach things, because it has provided a great competitor to do better surgery too.
You know, that's a great point, Mike, that you know, now that we're, I guess we're competitors, we think of ourselves as being collaborators, but now that, you know, there's so much new innovation that's going into TAVR valves, that I'm sure it's going to spill over into surgery as well. We're seeing significant changes in surgical valve technology as a result of TAVR.
I was just going to say, correct me if I'm wrong, but this leaflet, decalcified leaflet technology has actually, the material itself has been implanted already in humans for
Over 20,000 people, yeah.
Yeah, and so we have some evidence already that there is some durability, there is real lack of calcification.
So Becky, you're going to help us to understand a little bit more about valve leaflet physiology and how it relates to imaging, and why it's so important to be looking at hemodynamics and valve leaflet geometry.
Yeah, I think the beauty of this valve is its very long coaptation zone, but also the shape. I'm totally fascinated by the fact that the leaflets are indeed shaped like the normal leaflets. And so there's a couple of things that I think probably happen because of that shaping of the leaflets. One, the opening is going to be much more natural, much more like a native valve. But two, the closure forces may actually be less because it's shaped to be closed. And so it's opening with the force of the ventricle, closing kind of naturally, perhaps even with less force required to close it than in some of these other valves that we have. And so I'm fascinated by the shape of the valve and how that's going to really impact the hemodynamics. But I do think that if it does, and I think you've shown me data that it actually does have excellent hemodynamics, it's going to play a role eventually also in durability. So when we look at, we know that the surgical valve has a sewing ring, and so it's naturally going to be a little smaller, and we've shown that the prosthesis patient mismatch is going to be better for the TAVR valves, but hemodynamics matters. And so if you have low gradients, big valve areas, the leaflet stress is less, and my suspicion is that it will play a role in also durability. The DurAVR you can see on these images is just got very laminar flow throughout the stented valve portion of the valve, and it's really quite nice. I think it's going to prove to be a nice hemodynamics that we see in the end. And this is just CMR flow dynamics and work done by our friend Joao Cavalcante, and you can really see that in a normal healthy valve you've got this nice laminar flow with not a lot of turbulence, so there's not a lot of reversal of flow. It's just nice laminar forward flow, and that's really mimicked by the DurAVR. And so you can see that the flow dynamics is good, that there's not a lot of turbulent flow, which is going to affect the leaflets, maybe cause them to flutter, put increased stress on those leaflets, as you can see in some of the valves examples on the second row. And again, I can't help but think it's got to play a role in the durability of that valve as well, but also in how patients feel, in the regression of left ventricular hypertrophy, and how far they're going to be able to walk, and and all the functional and quality of life metrics that are so important to the patient.
Well, I think we're going to have to learn a whole new language here. These studies are really exciting, these CMR studies, and looking at flow dynamics and looking at opening patterns and closing patterns, I think is going to be very interesting as we go forward, and trying to understand the implications with respect to immediate hemodynamics and then long-term changes. Now, when we speak about coaptation length, we carry with us some excess baggage. There have been times when increased coaptation length has been associated with pinwheeling. When you think of coaptation length, you've got to think also about how do you size the device and what implications does it have with sizing. And maybe Chris can address those two issues.
Yeah, I think that's a great question, Marty. And I think the way that this coaptation, I think, works relative to the normal one, where it is the three pieces that are kind of now forced up together versus the shaping of the long, flat coaptation surface, I think there's some encouraging early bench work that demonstrates that it's likely lower leaflet stress and strain because it kind of smoothes out that contact forces across. But Mike, it appears as a surgeon's perspective of what are the implications when it's flat?
First of all, it's not coaptation length that leads to pinwheels. It's free margin length that leads to pinwheeling. Coaptation length is how much of the leaflets touch each other.
Right.
It's the free margin, and what it should look like, we know from aortic repair, is a pair of hands praying and not touching at the fingertips. And Becky will tell you when we do an aortic valve repair for AI, she wants to see have these hands praying with a lot of leaflet touching, and that's the coaptation length. And it matters a lot, and we certainly know from valve repair in both the mitral and the aortic that the length of coaptation is related to the success of that valve working over the long haul. It builds in a margin of error for you.
I could add also the fact that nature is beautiful. No?
Yeah.
So yes. we are born with the valve with such a shape, coaptation, which is good. And when we have this disease and the impact of the disease on mortality, we are all aiming to treat it. So for ages, the research have been able to deliver us some valves, which were fantastic because they saved lives. But they were not optimal, I would say. What we have learned from surgery, and this is very important, is that in younger patients below 70, these bioprostheses are not lasting for a long time. Yeah. And when it works, for example, when you do a Ross procedure, there is no doubt that this is one of the most important and beautiful surgery because you restore physiology. So what is missing, the gap is here, is try to have a new leaflet design that mimic the nature and the beauty of the nature. And I think this is, again, one of the chances that we have in our hands here with this technology.
I think the other feature here that's very interesting is that when you have basically this 3D geometry from a single leaflet design, far fewer sutures in terms of being able to fix this to the frame. And I think everybody knows, it doesn't matter how good you are, when you put sutures into tissue, it's going to have an effect on healing and on durability. So the fact that you're able to fix this to the frame with a smaller number of sutures, I think that by itself is going to make an important difference.
And that's a great point, Marty. This is just actually 100 suture bites actually roughly for this valve, where the traditional three-piece valve is about 800 suture bites. And so I think you're right, because also, we're all humans. We've all been to factories and seen these valves being made before. Our minds are blown. But to tell me that you can perfectly do it every single one of 800 suture bites every time, that's challenging. There is variability in these type of things, and there may be potentially implications for that too. So I think that is a really interesting point.
So, all right, well, we're going to continue in our journey of trying to understand a little bit more about the technology of valve leaflets. And I'm going to ask Thomas to talk a little bit about a surgeon's perspective. What turns you on as a surgeon with respect to a different valve leaflet design?
Thanks, Marty. So, TAVR is in its 20th year. And this was and is a chance for the patients. It's safe, it's easy. Yeah. And we have been generating tons of evidences that support what we are saying. But there are some gaps still, and we are working on that. So one of the most, I would say, important question today is, what is the additional value of TAVR, for example, in small annuli. In women, where we know again that, as I've said before, the surgical techniques are a bit more challenging. I mean, having a TAVR, which can be done under local anesthesia, is a chance for any patient here. But what is important is that we should not sacrifice for the sake of the results today, the chances of tomorrow. And what I mean in this, if I jump on the last slide of Becky, this laminar flow, which is a normal valve, looks very similar to what DurAVR is showing us using the MRI. And you look at the surgical valves, but also the self-expandable valve and the sapien valve, you see that you have this eccentric jet. This is probably one of the explanations why the durability has failed to be effective in this category of patients. So I want to share this couple of slides with you that have been done by our colleague and friend Janar from Vancouver. So if you look these hydrodynamic studies in this small annulus, 21 millimeter round fixture, you would see that the design of the leaflet itself has an impact. Look at the difference. So the same surrounding anatomy, I would say. The effective orifice area, if you use a sapien balloon expandable device, is 2.3. If you compare it with the self-expandable device, the Evolute one, 2.4. Look how large is the EOA with the DurAVR. This is this is probably why the laminar flow we see in real life would have an impact and should have an impact and will enlarge and extend the indications of TAVR in younger patients. If I go to the next slide also, this is in a slow motion video also, and we were focusing on the comparison between DurAVR and a similar valve, which is a Sapien 3. Look at the end of the video on the right part of the screen. Look how wide and extensive the opening of the leaflets. This is what a normal valve behaves and look like. This is probably why also this laminar flow is there. And if I finish by this one, because as I've been saying, today should not sacrifice tomorrow. And look, for example, here we have looked at the Sapien 3 hydrodynamics. EOA is 2, gradient is 11. You put a DurAVR in this same valve, and you could still achieve higher EOA and lower gradient. So I believe that mismatch is not a futile question. I believe that mismatch will have an impact in long-term survival. And this is why I believe that DurAVR is a chance for the patients.
Yeah, that's exciting, Thomas. And I mean, just looking at these pictures, you can't help but be impressed that there are visible, you can quantitate them, but visible differences in the opening pattern, the closing pattern, and the orifice area for similar sized devices. So you have to believe that that's going to mean something. And if at the same time you've also changed the leaflet tissue itself from the standpoint of preservation and character, that might add to certain differences over time. So, these are very, exciting, analytic models. Again, we're going to have to take these into patients, without delaying any further, this device has already been implanted in patients, and we're going to ask Mike Reardon to begin describing some of the first in human experience.
Professor Modine mentioned the Ross procedure as an aortic valve replacement. It's the only aortic valve replacement that brings you back to a normal lifespan because you're putting in a living valve with normal physiology. And so our goal as interventionalists should be to create a valve that's as close to the human valve as absolutely possible. So you want to maintain your coronary access with a short frame. You want near normal hemodynamics. You want commissural alignment. Oh, by the way, it should be easy, reliable, and dependable to put in. Those are all great things we've talked about, but the proof of the pudding is when we put it into people. And we've done that. We've taken a population of 13 patients in our early feasibility trial to do that. If you look at this average age is about 74 years, almost 80% women. Now that's important because that means we're starting with a higher risk population with smaller annuluses. And these weren't all chip shots. We had bicuspid valves, we had heavily calcified valves, we had heavily calcified annulus with an average annular diameter of under 23, just under 23. These are small annulus, difficult cases to start with. And when we did this, the clinical outcomes were no deaths, no strokes, no minor or major bleeding, no reoperation intervention, and no myocardial infarction, with a mean coaptation length of 8.3 millimeters. When we do aortic valve repair, we shoot for 8. That's an optimal repair. Well, this is getting back to a normal-looking valve that's been properly repaired. If we move on to the post-procedural outcomes, we have no device-related complications, no moderate or severe paravalvular leak, one mild, moderate central AR. This was a very early case. It was really a valve sizing issue. We've all gone through our valve sizing issues as we've moved through our TAVR world. We have one access site complication resolved on day 1, and one new pacemaker that had a baseline right bundle branch block and left anterior fascicular block. Question is why didn't this patient maybe have a pacemaker beforehand? So really good results. So what do we see at the echo? If these come back and Becky looks at these people, we see consistent laminar flow throughout the valve. We see native-like leaflet function. Becky already showed you the valve, how it opens and closes, and looks very normal. If we took a frame away from this, we might even fool them in the echo lab that this is a normal valve. We had an EOA, a mean EOA of 2. Now remember, these are valves that have an annular size that are just under 23, and yet we've got an EOA of 2, and that gives you an index EOA of 1.15. These are really excellent results, and it's getting closer to what we'd expect from normal valves. And certainly we know you need an EOA of 2 in our young people that want to be physically active. This will allow them to be active without substantially raising their gradient. And we look at single-digit mean gradients and DVIs above 0.5. So the hemodynamics of this valve in this early patient population are absolutely stunning. The safety and implantability of this valve for the first 13 cases is absolutely stunning. And certainly all of us hope that this is going to lead us into the future in a valve that's more like a normal valve that has the ease and predictability of the balloon expandable platform that we're currently using.
Thank you, Mike. That's really a great description of an early experience. I mean, we only have 13 cases. We have to be humble, but the reality is, as Mike said, bicuspids, relatively smaller annulus sizes. Vast majority are women with hemodynamics that good. Certainly is very, very encouraging. With any new device, there are things we have to learn. We have to learn about sizing. We have to learn about, I'm sure, many other subtle features. We want to make sure that we're getting better at aligning the commissures, although the geometry of this device, it's a relatively short frame device, I think, is going to favor not being a real problem with respect to coronary access. But this device has many positive features, and we're beginning now to see that borne out in patients. So how are we going to study these patients to be able to, we've already seen some differentiating acute hemodynamics. We've seen some echoes that look to be different in terms of coaptation length. What are we going to do next in terms of studying these patients to continue to learn about how these innovative changes affect characteristics that are going to be important to us?
That's a great question, Marty. So, I think one thing that I've really enjoyed is we've all partnered in working with the Anteris team to help kind of cultivate this. It's not a one person in a box coming up with ideas, but really a conversation of what are unique things our field should look at to try to make things different or better. Becky, we had the conversation the other day about the exercise stress echoes, which you're already doing. And we've seen really encouraging results that by exercising these patients, we don't even see gradients over 12, even at peak exercise in these smaller annuli patients. We've more recently talked with people like Stephanie Sellers out of Vancouver and Jannar that say, like you alluded to, this is early, it's very encouraging, but you're fooling yourself if everything's completely perfected by now. What are things we can keep looking at to keep refining, to continue to try to make this a little better and better and better? So Stephanie's looking at things and saying, how do these close? What are things we want to be thinking about in the future? Starting to already think about second generation, third generation, and continuing to, I think, work together to look for unique imaging modalities, looking at unique people from the bench to bedside perspective, to try to think about how we get closer and closer to mimicking a healthy aortic valve.
Although I can say we've already got techniques that we've been used that have been applied to a variety of different valves that will give you a good basis for comparison. Just looking at CTs at a month or at 6 months, with 4D time resolved CT, it's going to be an important test to see if this whole issue of valve leaflet thickening is different. with what we think are better hemodynamics in different tissue.
That's a great one. And we do have these data, and they are very encouraging. We have to continue to understand, obviously, optimal frame expansion. Joao's taught us all lately how important it is to make sure that you have symmetric expansion. These are also examples of things that can be identified to continue to optimize, of making sure you get as circular as possible and these kind of things. So I think it's just a continual learning process.
You know, one of the things that occurred to me when you have new technology like this, in terms of how you apply it to patients, of course, we can do some of the standard studies. Mike knows all about those. We can do a device versus device, this, that, or the other. We can pick subgroups and whatnot. You know, we can pick, you know, young, difficult patients and start thinking about what's going to happen in situations where we would expect accelerated structural valve deterioration and use that as a base population to look at. Or we could go the other extreme and take very complex valve morphologies and see if a better valve design from the standpoint of leaflet geometry and composition can make a difference in bicuspids or in certain other situations where there's heavy calcification. So there are a lot of ways, I think, that, you know, we can go to be able to study a device like this, which I think is going to be exciting for the future.
Yeah.
I think we hold on our shoulders the responsibility of moving forward permanently. So, I mean, you see, Mike, Professor Reardon have shown that 13 cases, no complications, easy to implant. This is one of the most important features that we need. Security for patients first. And then we look at the gaps. We need a short profile for coronary access. This is for tomorrow. And the durability. But we have to be very humble, as you have said, and very careful. We are generating evidences. But the fact is, we have something here. We're seeing that something different is happening, which could have a major implications on one of the most important features in the manipulation of the design and iterations of design.
I think if we could absolutely normalize the valve performance from the standpoint of hemodynamics and valve behavior, you know, relative to a normal aortic valve, that would be such an important advance. But I would also say that part of the success of what we've done with transcatheter valves is that we've always been self-critical.
Yes.
And we try to highlight the areas that aren't perfect. And in doing so, we force ourselves to continue to iterate. And, you know, it's something that I would suggest. I think there's been an incredible amount of interesting thought that's been poured into this design. But I think over the next many months, early years, and numbers of patients, you're going to see things, you're going to learn things, and having astute people like our surgical colleagues and Becky looking at all these images, you're going to get a lot of recommendations on how even this design can be iterated and applied in a way to try to take optimal advantage of some of these design characteristics. But that's part of the excitement of where we are.
I think you said it so nicely, Marty. That's the fun thing, at least I'm having so much fun with all you guys in this journey together on this, is that it's not, we're not just putting our heads down and 2 years later looking up. It's constantly, hey, Becky, is this really, I mean, the first time Becky saw the laminar flow, she's like, I need to see more, Chris, I need to see more, right? So, all right, we went back. Of course, I've never asked Becky for something, or I showed her something and it was immediately good, but now she sees the MRIs and she's fully agreeing now too. And so I think this is the process though. You have to have real feedback, and this goes to everything we're seeing at this meeting, right? If you just get excited and you hide in your silo and you just tote things around for a few years, you don't advance things. You have to move the field, and this is how, this is an important thing for all of us, is when devices get ready to finally make it to commercialization, it can't be the device we need 5 years ago. It needs to be the device we actually need 5 years later. And so I think with these effective partnerships that we have and many others, this is the strategy of the real relationship of a company, engineers, physicians, that really know what's happening, to try to work together to really develop something differently. And I think ultimately that's how most things end up really helping revolutionize and change our field.
And I also think that we're very fortunate in this country to have regulatory agencies that are willing to work with us.
Yes.
And would allow us to do some creative things. And we should take advantage of that from the standpoint of trial design, from the standpoint of mechanistic sub-studies, doing comparative studies. I don't think we want to spend the next decade or two just repeating the same kinds of clinical trials that we've done in the past. So it's incumbent upon us to be able to hopefully give you the kind of good device to allow some of the features of this technology to be best demonstrated with some innovative imaging or study design concepts. So that, again, gets back to how we can work together.
Absolutely.
So this has been an exciting discussion of a new technology that we all think has got great promise in being able to take transcatheter heart valve approaches to yet another level. So nothing is static in this field. It's
Very exciting, Marty. It makes me wish I was 40 again so I could watch this progress.
I know, Mike. I share your frustration that some of the fun, we may not be there to see it actually pan out. But certainly, it's been great to be involved with so many creative people and to see these continued innovations happening. Thomas, any final thoughts?
It's a fantastic journey that we are living here. And again, without research and innovation, everybody will not have the same chances.
Yeah.
And we're doing this for us, but also for the community. And this is one of the most exciting projects that we have been living now.
Yeah, and think about how our patients are benefiting from this.
Yeah.
I mean, that really is, the end result is that's why we're all here.
Yeah.
The ability now to even to think that we could treat younger people with bioprosthetic valves with the hope that it might be the only valve they'll ever need with hemodynamics that simulate a normal valve, it's pretty mind-boggling.
Yeah, I think even the valve-in-valve aspect of
Right.
this particular hemodynamics is
Right.
pretty enticing. That and the small annulus. I think
Yep.
we've been dealing a lot with small people, small women, and this is just it's a valve for everyone, including the patients that have to have a repeat valve-in-valve.
Well, this has been a fun conversation. I think we've really dug pretty deeply into a new technology, looked at some of the ex vivo studies that are intriguing and different from the standpoint of mechanistic insights. We've looked at some of the first human use experiences, which are quietly encouraging in terms of being able to validate that some of the design concepts may have clinical relevance. And now we're all anxious to see what some of the next steps are. So I want to thank Chris and his group of scientists and engineers at DurAVR because this is one of the hot new areas, and we love having an opportunity to showcase this at a meeting like TCT.
Thank you, Marty.
Thanks, everybody.
Thank you very much.
Thank you.
Thank you.